أحب أرحب فيكم جميعا اليوم. أهلا بكم ومرحبا جميعا وفردا في الحلقة الأخيرة من المناعة في زمن الكورونا. معنا ضيف عزيز علي وعلى كل خريجي جامعة مانشستر وصديق شخصي لي أنا، الدكتور جون ورثينجتون من جامعة لانكاستر. حيتكلم الدكتور جون في دقائق قليلة أو بعد دقائق قليلة حيتكلم عن نفسه، حيعطيكم فكرة عن إسهاماته العلمية ومشاركاته. رفيقتي في هذا اللقاء الرائعة والمتميزة دائما الدكتورة طرف التركي. Okay, so John, do you want to tell us a little bit about yourself? You're in Manchester now, aren't you?
Lancaster, yeah.
In Lancaster.
Yeah, so thanks very much. Yeah, so I met Mushraf and Tarfa when I was at Manchester University, and I did my PhD there under the supervision of Professor Richard Grensis, who's a real kind of trailblazer in the world of kind of helminth infection. And it was also working with Professor John McLaughlin, who's a clinician and a gastroenterologist. So it was really looking at the kind of crossover between parasites and also the endocrine system, because John was very much interested in the regulation of feeding and nutrition and the molecules and the hormones that drive satiety. And I'll speak about that a little bit later in my sort of talk. And then following that, I did a couple of postdocs in Manchester with Mark Travis, which again, I was looking at inflammatory bowel disease and parasite infections, always kind of focusing mainly in the gut and the kind of regulation of these pathways. And then finally from that, I was able to gain a Stepping Stones Fellowship at Manchester, which led me to be able to establish my own lab and gain a lectureship at Lancaster, which was about coming up for a kind of 4 or 5 years now. And I've been there since and kind of enjoying my time there, but I still have kind of close links to people at Manchester and also with Mushraf as well.
Yeah, you're still doing, I mean, your project is roughly around, I guess, fat cells as well, satiety and regulation as well. So you're following in the same footsteps you've been going.
Yeah, so exactly. My kind of PhD was kind of understanding these cells a little bit. And then with Mark, I went to do kind of more, I suppose, hardcore immunology in the gut with regulatory T-cells, the breaks of the immune system. And then after that, I was always kept in touch, as you always do, with the area that your PhD was focusing on. And I knew there was just so many more unanswered questions still on these kind of chemosensory cells in the gut. And so it was really nice to kind of be able to go back, and we've started to ask some of those questions now and started to get some quite interesting answers, which I'll touch on a little bit in this session, but mainly I'll be talking about published work today, but I'm happy via email to answer any questions following on from this.
Okay, but just before you go, one important question. How's the weather over there in the UK?
It's ridiculously nice at the moment. Yeah, it's really sunny, even in Manchester, which I suppose should be a hint that something is wrong in the world.
Maybe all humans should stay in. Is that what you're saying?
Yeah, well, no, that's maybe.
All right, John. I believe you want to. You've got a presentation.
Yeah, so it'll just be about kind of half an hour, just going on how my PhD became interested in these cells, which some people might not have heard of, at least in the relationship to the immune system.
Okay, the screen is yours, my friend.
Okay, so today I'm going to be talking, or this evening I'm going to be talking about the kind of immune endocrine interactions, specifically at the intestinal barrier, but a lot of these interactions also happen at other barriers, so especially things in the lung. We've got lots of kind of chemosensory apparatus there and throughout the body. And things have been kind of found within the thymus very recently as well in terms of these kind of tough cells. So I've always been interested very much in the kind of intestinal immune system, in particular this kind of balancing act. So we have someone here on this kind of high wire. If you walk this thin line in between, you're fine, but if you can go kind of fall off in one of two sort of ways. And the reason for that is that the intestinal epithelium is obviously an essential point of absorption for nutrients. So its main role is, during feeding to absorb these key nutrients for survival. But it's also got to act as an efficient barrier against obviously toxins that can come in through the oral route, or also kind of microorganisms, so infections, but also the huge microbiome which is in our gut is obviously essential for digestion. So it's kind of no wonder really that the gut hosts the largest immune system of the body, and it's essential in determining kind of this tolerance where we don't want to react to things which aren't harmful versus kind of immunity. So it's that very fine balancing act. And so if this kind of balancing act fails, you can, as I say, fall off in kind of one of two ways. You can end up with autoimmunity. So here we've just got a colonoscopy, and you can see on the far left kind of a healthy gut. In the middle here, we've got kind of inflammatory bowel disease, which is where you make too strong an immune response to your microbiome. So you're basically overreacting to things that are essential for your body to function. You get this kind of really nasty inflammation. Alternatively, you can not make a strong enough immune response and can end up with chronic infection. So here you might be able to see just on the far right, we can see some small little white worms, which are kind of trichuris, which was this kind of whipworm, which can inhabit the gut. And this was found on a kind of colonoscopy. So we've got this kind of fine balancing act where we want to be somewhere in the middle, you know, as with most things in life, you want to have that kind of fine balance. So the kind of thing I was interested in is this kind of layer of epithelium, which acts really as the kind of sentinels of the gut. So they divide the huge amount of microbiome, food, nutrients, and potential kind of pathogens from the kind of largest immune system in the kind of lamina propria, which, although there is obviously the mucus layer, you've literally got this kind of one cell thick barrier, which is preventing these two things from coming into contact and causing inflammation. So the epithelium is made up from these different cell types, which all arise from this kind of pluripotent stem cell, which is located in the crypts of the gut. And these can differentiate by different transcription factors, which we now know a lot more about. They can become the absorbative enterocytes, the goblet cells, which produce that important mucus layer by producing mucins. We have kind of Paneth cells in the small intestine, which migrate down, and they produce antimicrobial peptides and also help the kind of stem cell niche survive. We've also got Tuft cells, which were a rarer cell population, but have become really of great interest recently because they've been shown to produce alarming cytokines such as IL-25, which can drive immune responses to produce the T helper, well, the type 2 immunity, which is very important in expelling these kind of helminths. But there are also other rarer types of cell types, and the one I became interested in during my PhD was this endocrine cell. So as I said, they're very rare. They make up about 1% of the epithelium. They're very spaced out. They have a lot of taste receptors on them, but collectively they actually are the largest endocrine system in the body. So although they're very rare cells, there's a hell of a lot of them all together, and they do a very important job. And what they've done, what they've classically been studied for, is their roles in detecting luminal nutrients. So they basically can detect a lot of different things via G-protein coupled receptors. They're kind of like little taste buds in your gut. What happens is they will sense what nutrients are available, and then depending on what they've sensed, they can then produce different peptide hormones. And these are things such as cholecystokinin or glucagon-like peptide 1, which I'll kind of mention later in this talk. But they can produce a variety of different hormones. And the key thing is, from this, they are able to signal via kind of vagal afferent nerves to the brain and cause basically the ideal situation to cause digestion for whatever luminal nutrients they've sensed. In other words, it's a really nice system of being able to detect things and then respond appropriately. So as I said, these vagal afferents can signal to the brain, and they also result in the feeling of satiety. So in other words, they control when you want to stop eating. So for example, things like cholecystokinin or CCK, it's produced a lot in response to very long chain fatty acids. And so what it does is it basically makes you feel full, it gives you more time to digest them, it closes off stomach sphincter, so you basically have less food going in, gives you more time to dissolve these things. GLP-1, glucagon-like peptide 1, is a lot more involved in the control of glucose, so it does a lot of insulin secretion to basically control your blood sugar levels. So basically these cells have classically been known to see what you've just eaten and produce the ideal response via these kind of taste receptors. But importantly, the thing I was always very interested in was, under epithelium, as we said, you have the largest immune system in the body. So kind of my lab looks at the kind of important question, at least that we think, is what are the interactions between these intestinal, endocrine and immune cells? And that kind of comes in three different types of questions. So how do immune cells communicate with these endocrine cells? How do the peptides that are produced influence the barrier? And then finally, is there a possibility that these peptide hormones could act almost as cytokines and communicate with immune cells of the gut themselves? So I became interested in this originally during my PhD, which was based, as I said, by Professor John McLaughlin, who's been very influential in studying cholecystokinin. And what they tended to do was they would measure levels of CCK in blood serum from healthy volunteers after a controlled meal. So they would normally eat soup, which is very good in making you feel full, and see the levels of CCK that were produced. And they had a healthy volunteer, and he was actually a professor of gastroenterology at Manchester in Salford, but I won't tell you who he is. But he's now retired. But he took part in the study, and they were looking at the plasma CCK levels after this kind of test meal. And interestingly, his fasting CCK was kind of 4 picomolar, which was compared to the kind of 1 of healthy control. So he had 4 times as much of this CCK level in his serum. And then after he'd eaten his meal, it went up as expected. But again, it went to super high levels compared with these kind of actual healthy controls. So he became quite worried that he was potentially had like an endocrine tumor of the gut and was kind of quite panicking. But it turns out he'd actually just returned from a conference in China, and he was reporting nausea, anorexia, kind of bloating, and also had lots of fat in kind of his stool. And it actually turns out he was then diagnosed with Giardia, so he wasn't a healthy volunteer at all. He had this small intestinal kind of parasite, which is, from all accounts, not a nice thing to have. And so that led to a study by Fiona Leslie in collaboration with John McLaughlin, where they looked at upper gastrointestinal infections and basically wanted to see how the CCK levels changed. And as you can see here, what they saw was that compared to the uninfected proper healthy controls, where their fasting levels of CCK rose slightly just after the meal and then returned to normal. Patients with upper GI infections, their levels were significantly higher than controls and raised to really significantly higher levels 15 minutes after that meal. So there was, for some reason, these kind of satiety signals were really increased. And that's kind of, when you look back in the literature, it is known. We know that gastrointestinal inflammation is often associated with hyperphagia, so that's the reduction in feeding, and also weight loss. At the time, the cellular molecular basis for this was unclear. There was kind of the idea that, which is sometimes the case when you get high levels of cytokines within the serum, that can signal directly to the brain or sometimes via the vagus nerve, which also signals to the gut. But there was a lot of kind of unstudied areas here. But we knew that these endocrine cells were implicated. As I said, we saw CCK increases in humans, and also in kind of veterinary science, when people are looking to have obviously larger animals, they want them to eat more. There've been a lot of studies with infections, and they could see that CCK went up, for example, in livestock, lambs and pigs, when they're infected intestinally. And as we said, also in that human study. So what we wanted to do in my PhD was kind of have a model that we could use and investigate, something that we knew what the immune system did and it was controllable. And this is when John McLaughlin teamed up with Professor Richard Grentis at Manchester, and started to use, he's an expert in parasitology and immunology, use a good model. And we started to use this thing called Trichinella spiralis, which is found in undercooked pork or kind of different animals as well. They can infect most mammals, including humans. So always had to be always a bit nervous as a first kind of year PhD student when you're working with something that can infect yourself, but it's kind of fine. It's, you know, unless you drank the sample, you would be fine. And what happens is these kind of nurse cells that form in the striated muscle, they reach the acidic pH of the gut, they hatch out, and these newborn larvae enter the small intestine, and they form these syncytial tunnels within the epithelium. They then mate, produce more newborn larvae within the small intestine, which can then migrate via the lymphatics in the blood. We're not too sure how that happens. And then they find striated muscle and form nurse cells again, and so the life cycle can continue. And with very high level doses, you can actually cause quite bad pathology. But people who do have trichinosis, they tend to have kind of a rheumatism feeling, and there's no real drugs once it's kind of there that can really control that to a great extent. So in the mouse models, we knew that there was a well-characterized transient Cd4 helper T-cell response. So these Cd4 helper T-cells could really produce Th2 cytokines, things like interleukin 13 and interleukin 4, and drive inflammation in the small intestine and expel the worm. That was already linked with goblet and Paneth cell hyperplasia. So we knew that other secretory cell types did increase during this inflammation with this parasite, and that causes the adult worms to be expelled from the gut. And with Trichinella, it's very much mast cell dependent. So if you block mast cells or some of the factors they produce, there's a real delayed worm expulsion. What we know happens is mast cells degranulate. They produce mast cell proteases, which basically break down the epithelial tight junctions, and that allows fluid to leak into the gut. Th2 cytokines also produce contraction of the gut, increased mucus, and it really flushes out the worms from the small intestine. And then that basically is protective. So my kind of important question here was, is the adaptive immune response that we see and we already knew about during Trichinella infection, was it influencing these endocrine cell function? So first of all, we wanted to see if, as we saw in humans and lambs and pigs and things, did CCK actually change during Trichinella? You know, every infection, you've got to remember it could be different. So here you can just see, this is a villus here, and I've stained for CCK using an antibody and done some immunofluorescence. So you can see these cells are quite rare, just 3 here on this one villi. And in the naive state, you know, around looking at 20 villus crypt units, so, you know, looking at 20 separate kind of villi, we only saw around 3 or 4 cells, so they're really rare, these cells. But following infection at day 6 and 9, which is the kind of height of the inflammation in this infection, we could see that CCK cells did increase significantly. And then once the worm was expelled around day 12, still slightly raised, but it kind of returned to normal levels. And that actually, when we looked at the weight loss, because CCK obviously can drive feeling full, we looked at the food intake, and you could see that as the CCK increased, the mice reduced the daily amount in grams of the pellets they would eat. And that kind of, again, mirrored the changes in the CCK cell number. So that was very suggestive that CCK was responsible for these changes in feeding. But we then used, briefly, we used a CCK knockout mouse that we got from Linda Samuelson in Michigan. And when we infected these mice, there was basically no alteration in feeding. So it seemed that this one peptide hormone, CCK, was causing these changes in feeding. So the next question is, you know, what happens with the kind of immune response? So we used the SCID mice, which has no B or T cells. In other words, we're saying when we don't get that T helper response driven by the Cd4 T cells, do we still see changes in CCK cell number? And when we looked here, so again, going back to this first graph, in SCID mice where T cells weren't present, we could see there was no hyperplasia of the CCK cells, no increase in those satiety hormones releasing cells during inflammation. The worms were still there, they were causing damage, but there was no increase in those CCK numbers. And importantly, looking at the weight, the amount of food that they ate, again, there was no change here. So we lost that increase in CCK cell number, and we saw no changes in the feeding. But again, just to make sure that it was the T cells that were responsible, we did adoptive transfer. So we purified T cells from previously Trichinella infected mice, put them into the SCID mice, and to see if we could rescue that phenotype. And what we saw, again, was not quite as pronounced, but we did see significant increases in CCK cells on day 6 when we put the T cells back in. And again, that mirrored a slightly reduced reduction in feeding, a double negative there, sorry. But basically, you can see that when you do put the T cells back in, you also get the increase in CCK and the reduction in feeding in the mice. So that basically told us that CCK hyperplasia and the hyperphagia it drives is under the control of the adaptive immune system. So that was kind of really nice and interesting. But my question, sort of as I advanced in my PhD, was kind of why is the immune system hijacking your feeding pathways? So it could just be a kind of an innate response in a way. Obviously, if we've been eating something from a contaminated area, going back evolutionary, it will be advantageous to basically not want to eat anymore, because then you would reduce the load of infection that you were taking in. But at the time, I was looking at a lot of papers from Graham Lord, who is actually now at Manchester University. But he'd done a lot of work in London looking at leptin, which is a real link between nutrition and immunity. So leptin is produced from adipose cells, so fat cells, and basically the amount of circulating leptin you have is normally proportional to your adipose tissue mass. It's actually structurally similar to members of the long-chain helical cytokines, so things like interleukin-6. So we know, again, something in endocrinology, which actually does more of a kind of cytokine job. And Graham Lord had shown really nicely that it drives a T helper 1 immune response in T cells. In other words, it drives a response traditionally more suited for kind of intracellular pathogens versus these helminths, which require that Th2 or type 2 immunity. So that hadn't been done, that was all done in splenocytes. I wanted to see if that happened in the gut, because again, it's always really important to work on the tissue where the niche is of what you're working on. And what we can see here is that we purified Cd4 T-cells, and I pushed them towards a T helper 2 state. In other words, it was like if a T cell from the mesenteric lymph node, which drains the gut, was making a Th2 response, what would happen when we put leptin into play? So a kind of in vitro kind of helminth infection, if you will. And complementary to Graham's work, when we added leptin into the system, we could see that the normal low levels of gamma we saw, because obviously I pushed it to a sort of Th2 response, were actually significantly increased. However, when we looked at interleukin-4, which is a cytokine which is produced at a very low level, it's a very potent cytokine. Within the Th2 polarized cells, we could see that T cells here, just on flow cytometry plots, were making around 6%. But if we added leptin in, it really knocked the levels down by about a third. So again, fitted with the kind of Graham Lord's previous work. And that kind of made me kind of take the next step, where it was like, okay, adaptive immunity might be altering feeding pathways to alter the levels of the adipokine leptin, and that might actually help drive a Th2 response. In other words, your immune system could be altering your weight to be beneficial in terms of fighting off what kind of pathogen you have. So the question was, if we restore the leptin, do the worms stay? So first of all, we again infected some black 6 mice with Trichinella, as we had done previously. We had to first of all, again, check that leptin did go down. We knew the mice lost weight, they ate less, and they also had a visible reduction in the fat pads. So we would hope it would go down, but obviously you always have to check. And again, what we saw at day 12 post-infection, when they'd gone through that period of reduced feeding and weight loss, the leptin had significantly dropped. So what we wanted to do was kind of bypass that weight loss. So we kept the leptin levels the same with twice daily injections. And when we did that during the period of weight loss, we could keep the leptin levels the same as basically a naive state. So although the mouse was losing weight, the leptin levels we maintained throughout the whole infection. When we did that, we looked at cytokines. So with the leptin treated mice, just in white, these are all infected at kind of day 12, I think. Gamma, there's a slight trend, but it was very noisy. There wasn't really much difference. When we looked at the levels of Th2 cytokines, if we kept leptin the same, in other words, it's as if the mice weren't losing weight during the infection, we saw a significant reduction in IL-4 and also IL-13, so those key type 2 cytokines. As I mentioned at the start, mastocytosis was essential for kind of expelling the parasite, and we started to look at different mechanisms. And we could see that in the leptin-treated animals at day 8, so we saw an initial mastocytosis began as normal, as in kind of PBS-treated infected animals. But when we kept the leptin levels the same, we never really saw those high peaks of mastocytosis. It was a significant reduction when leptin had come in. And most importantly of all, when we treated those animals, we could see that there was a big delay in the expulsion of the worm. In other words, the adaptive immune system was driving CCK feeding pathways to mediate helminth expulsion. So that was kind of a really nice kind of study, which has actually been taken forward a lot by other groups, and they've done things with viral and bacterial infections where they've looked at alterations in weight loss. And you can really, it goes back to the old adage of kind of starve a fever, feed of cold, starve a fever, which is a kind of old adage which might have some truth in, and now has been kind of shown quite nicely. There was a real nice paper from Metzitoff which took this looking at virus and bacteria as well. So first, the conclusion here is just to say we can see that the adaptive immune system can signal to these different endocrine cells, and we know this is by interleukin 13 as well. It causes a hyperplasia of these CCK secreting cells and a hypersecretion, and this can feed back on feeding pathways and alter leptin levels, which then kind of allows an increase in kind of the Th2 response to expel the worm. And we published this a good few years back now. Because it was to do with weight loss and worms, it kind of got a bit of interest. So I was kind of shocked one day after chatting to someone on the phone, and it ended up being on the front cover of the BBC News webpage, which was kind of very flattering and things. And I remember kind of phoning up, I think I phoned up my mum at the weekend to just sort of say, oh, we got this kind of thing. It's quite nice. Thought she might be a bit proud, but in true kind of Andy Warhol saying you're only famous for 5 minutes. I think the same week, my uncle, who is an auctioneer, had been on a BBC programme called Celebrity Bargain Hunt. So there you go. This is with Miley in class, who's a pop star, and I think she's an opera. I literally had 5 minutes of fame, and then my uncle took over from me.
You know why I'm laughing? Because I remember that happening. I was in Manchester. Yeah.
I'm not bitter though. Okay, so then after my PhD, I went to work in the newly established lab of Mark Travis, who had been at Manchester and gone to San Francisco to work on integrins and moved back to Manchester. And we did a lot of really nice work in two postdocs where we were looking at the activation of TGF-beta, which is obviously a really important suppressive molecule of the immune system. We found specifically this molecule alpha V beta-8 integrin on different cell subsets in the gut. So we saw dendritic cells and Tregs, and we were able to knock it out and find some really nice kind of pathology and do some sort of nice kind of work there. Mark was obviously really encouraging and kind of helping me start my own lab as well. So in that kind of area, I was always kind of still interested in these endocrine cells and immune crosstalk because there was so much happening. And so at the time, we kind of knew that pathogens, and endocrine cells could also sense pathogens into, they had functional toll-like receptors. What they made in terms of cytokines and the hormones altered if you stimulated with them nutrients and pathogens. And they were also then found to have really, well, they're the main expressors of GPR43 within the gut. In other words, they're the key cells that can sense the short chain fatty acids that your microbiome makes. So I was really interested to get back into looking at these cells to see what could kind of happen. And so we've been doing a lot of work with inflammatory bowel disease and CCK, which I won't talk about today, but will hopefully be coming out soon. But another molecule that I'd just like to touch on is this glucagon-like peptide 1. And GLP-1 really came to the forefront of interest. Again, it's another peptide produced by endocrine cells. And it's normally produced rather in the small intestine where CCK is made. It's normally further down in the gut, or in the sort of distal small intestine and the proximal colon. And what people found that was really interesting is that patients undergoing kind of resections of their bowel, so when they had their guts made smaller, very obese patients, to try and help them lose weight and control diabetes, they actually found in some cases that the type 2 diabetes was cured overnight before any alteration in weight. In other words, their kind of insulin levels normaled off very quickly. The patient was still morbidly obese, but something was happening very, very quickly. And it turned out that GLP-1 was a potential factor in this. So normally, if you think the GLP-1 sensing cells are very far down the gut, by the time the nutrients reach, there's not many left, and you get a small amount of GLP-1 released. However, after this bariatric surgery, suddenly cells releasing GLP-1 are seeing a huge amount of nutrients as they've literally physically been moved further up by the surgery. And what was discovered that was GLP-1 is actually anti-apoptotic for the beta cells in the pancreas, so it kind of all made sense. People then really started to use the use of GLP-1 agonists, and this is where it goes a bit kind of science fiction-y, but it's true, believe me, when I first read it in a paper. But there's something called Glea monster, which is the name for this lizard here, and in its saliva and part of its bite, it produces a lot of alterations in hormones. And I think the idea is it's kind of like a poison. It alters its prey's endocrinology. They will then basically go into a sort of coma afterwards. They'll be able to catch them and eat them. So it was kind of this thing, you harness the saliva of a Glea monster, which is it's called, to potentially cure diabetes. And that was exactly the case. So if you take GLP-1 receptor agonists, you can improve diabetes. But interestingly, in type 1 diabetes, this can be done without the need for immunotherapy. So basically, you have immune responses to your beta cells. You add this agonist, you can make them survive for longer, but your immune systems would still be killing them, but it seems to have a cure. So that suggested that there was kind of some immunotherapy, there's some sort of immunoregulatory role of GLP-1 as well. So my other question, which we're also working on at the moment, is can GLP-1 directly influence immune cells? And I won't talk too much about this, but I worked in the lab down in Cambridge with Fiona Gribble, and at the time there was no really good GLP-1 receptor antibodies, so I was able to utilize a mouse that Frank Ryman had made, and that's a reporter mouse. So any immune cell fluoresces GLP-1 receptor. You can see in the pancreas, there's lots of pancreatic slice, you can see lots of GLP-1 receptors. So it was a really nice way to say, okay, do any immune cells have the receptor for this hormone that could be influenced? So we took the GLP-1 receptor red fluorescent protein mouse, we isolated immune cells looking at CD4 T-Cell leukocytes and wanted to see what immune cells could be found. And basically what we found was that there was intraepithelial lymphocytes within the small intestine specifically. So we don't see any in the large intestine, but we do see them in the small. And it kind of makes sense that these immune cells that sit right between epithelial cells where GLP-1 is released have the receptor for one of these peptides. And we went further down and we could find that it was the kind of gamma delta, so the sort of non-classical T cells, which we find a lot of at mucosal borders, and they have the kind of CD8 alpha groupage. So just to cut the story short, we're doing a lot of work with these at the moment, and again, we've got some quite nice results, which we'll be kind of releasing soon. This is actually when we discovered this, literally a few weeks later, this was also published by, his name has gone by Dan Drucker in Canada, and I had contacted him just previously, and he did let me have the GLP-1 receptor knockout mice. So although I was kind of scooped a little bit by him, and to be fair, I got into it through reading his papers, so he was obviously going to be onto it before me. But he's a really nice guy and has been really good with kind of reagents as well. So we're looking in the GLP-1 receptor knockout mice at a lot of different diseases. So hopefully I've kind of given you a bit of a flavour that, you know, the immune system can link with these endocrine cells. Some of the work I've not showed today where they can kind of influence the barrier, and also how they can potentially, certain ones such as GLP-1 can act as a cytokine. And just to give you a bit more kind of flavour of this, if you're interested in, we did this review in mucosal immunology a while back, and you can see now that we're really starting to reveal lots of different things that these kind of hormones can do. They can strengthen those tight junctions, they can communicate with the stem cells, and as we said, they might be able to direct cytokines directly. And there's a lot of information, some of them small papers. We need to see how true all of this is, but there is information suggesting that these hormones can directly influence immune cells. But for the IELs, not just because I've shown it, but because it's been very well investigated by Dan Drucker as well, that this is definitely happening, and we don't really see these cells with this receptor in any other barriers. We've looked in the lung, the skin, and it seems to be very specific to the small intestine, at least at rest. We've found some other interesting things, but I won't tell you that today. So I'd just like to, hopefully I've not gone on too long there, where hopefully I've given you a bit of a flavour of these endocrine cells that you might not have heard of before. And we've got that review if you'd like to find anything more about them. But I'd really just like to thank, obviously, John McLaughlin and Richard Grensis, Linda Samuelson, Fiona Gribble, Dan Drucker for the kind of reagents there. Mushraf as well for his kind of support through the process. I already had this on the slide, actually. I've just noticed that. It's a good job it was there. And then people who've done the work in the past, so that's people at Manchester. We've had lots of master's students and undergrad project students, and then also at Manchester, and then PhD student Alex, Adora, postdoc, and Kevin, a master's student at Lancaster, who've done, especially Kevin has done a really lot of work on the GLP-1 story as well. I'd just like to thank you very much for listening, and I'm happy to take any questions on this, and then we can move on to the kind of COVID area. Okay, thank you very much.
Thank you, John. Thank you for the very informative introduction and a brief on what you're doing. I should know this answer, actually. Interior endocrine cells. Do we know of any of their links with innate immune receptors, RIGs, toll-like receptors and whatnot?
So, so they do have toll-like receptors, and we saw some on the CCK positive cells. We saw they express TLR2 and 4. People have shown that they respond to that, so you can stimulate. These are kind of stem cell line studies. So you've got something that's called an STC1 cell, which is a model for these endocrine cells. And if you have those in addition, you give them nutrients, they secrete the different hormones as you'd expect. But interestingly, if you put LPS on, they will secrete hormones in different amounts, but they will also produce kind of cytokines as well. So yeah, they definitely can respond to those things. People haven't looked so much in terms of non-membrane sort of receptors, but that would be something interesting to look at.
We've got, we don't want to dwell too much on this topic because we've got so many questions, but I've got a colleague, Marcello Albanese, wants to ask you a question about this.
Yeah, sure.
Hi.
Oh, hi, Marcello.
Hi, can you hear me?
Yes.
Yeah, I can hear you well. Hi, Marcello.
I'm really bad dressed, so I'm not gonna switch on my webcam. I hope you don't.
That's fine, Marcello.
Well, that was very interesting. I would have a couple of questions on your PhD project. So
yeah.
The first one, did you have the chance to use the WSH mice that are knockout for mast cells in your model?
No, so this was all done in kind of it was published in later than we did it, so it was done in kind of 2008. So I think the mouse wasn't quite up to speed, or at least we didn't have any access to it at that time. But no, we didn't do anything with c-Kit because we knew that we could potentially block with c-Kit, but that would also alter cells of Cajal, which would reduce the kind of contraction of the gut as well. So it was basically, we decided not to go down that route. But yeah, it would be nice to kind of revisit that and kind of, use the nice mast cell knockouts and sort of see if that was still the case.
Yeah. And the second question might be a little bit naive. So I think these same cells that you presented are a target for an autoimmune syndrome if you knock out the AIRE gene. So,
yeah.
And I think there are mouse models for that, but I don't know. I mean, if there are some mouse models for that, first of all, and the second, if you had the chance to use it in your system.
Yes, you're exactly right that, you have that syndrome and there's mouse models for that. But the thing is, when you lose those cells, I don't know if anyone's made a kind of a model where a temporal model where you can inject and knock out the cells. But when you lose basically all your endocrine cells, there's massive problems, in terms of inflammation as well. And I think even if you had that kind of temporal, you could inject tamoxifen or such and then knock out the cell, I think just all cells, you'd still have lots of problems. But that kind of links as well as to why those patients have such alterations in inflammation and
yes,
kind of barrier function.
Okay.
Yeah. So what we've been doing is kind of going for the more specific approach where we can have GLP-1 receptor knockouts and knock out just that receptor on the immune cells, or knock out CCK. We've had to still use the global CCK knockout, as the kind of Billing-Cree system doesn't really knock it out that well in the small intestine, unfortunately. But, with the kind of CRISPR-Cas things, I'm sure we'll be able to get some nicer models soon to look further into that.
Yeah. Okay, thank you. That was nice. Thank you.
No problem, thanks.
So, John, go back to the topic at hand, if you don't mind.
Yeah.
So, 5 challenging questions that we still haven't answered in terms of our fight against COVID-19. Now, you provided the topics. So, if we would go through the first topic you would like to discuss, where would you like to start? What's the first thing that you want to present to us? What's the first challenge?
Yeah, so something I've been kind of thinking of a little bit, we have at the moment, there's a huge amount of calls going out for kind of COVID-19 research. And, first of all, I will say I'm not a virologist in any way. I'm very much gut-based, but we'll come on to that in a little bit. Is you kind of think, well, and you kind of hope that, maybe this will all kind of blow over kind of soonish. And also as a kind of someone who very much works on mouse models, I do use human tissue now, but, my history is all on mouse models. You kind of think, we'd have to get in certain specialized mice, which would have the ACE2 receptor, so it could be used. But as we're now seeing that things are really, we are in this for the long haul, whether we like it or not. We've been starting to think about those kind of questions of what would be quite good models to use and kind of what would be good in terms of the kind of mouse studies. And then we can really kind of, knock out certain cells and kind of do things a bit more. Obviously, the vaccination tests, which are happening literally yesterday on, people, are fantastic. But again, efficacy only allows you to do so much, well, if we have a good solid mouse model or any other kind of mammal, we'd be able to answer a lot more questions.
But we're
Sorry.
struggling with that, aren't we? With the mouse models. So as far as we know so far, some of the early papers that came out early in the pandemic, Bao et al. Was saying that we struggle with wild type mice where they don't develop a pathology at all in most cases. And then transgenic mice don't, if they do develop a pathology, it's somewhere different in its severity than humans.
No, exactly. So the first thing obviously we've got fantastic array and a toolbox of things to use for the mouse, so that's why people tend to go for that. But yeah, obviously with your basic kind of different background of mice, they're just not really getting infected, so people then went for immunocompromised, rags and skids and things to see if we could work a way around that. But again, no such luck. And we now know that COVID-19 uses the same receptor as SARS, so it needs that kind of the ACE2 expression to get into the cells. And because mice have a very different molecule, if at all, I have to double check, we have to use mice where they have the ACE2 expressed on them, and kind of the best thing to do is, rather than overexpressing it throughout the mouse, because then you get this kind of super susceptible mouse where you've expressed ACE2 in high levels in all tissues. What the people have done the best way is expressed the human ACE2, but on the mouse ACE promoter. In other words, you get it where it should be, kind of in high levels, which is in places like the lung and the gut as well, which we'll come on to.
But they became extremely, extremely ill, these mice that express the humanized ACE2.
Yeah, so I think when they overexpressed the human one throughout, so you just got super high levels. They obviously put it on a stronger promoter, I'm not sure which one. And then because it was so high, the mice really succumbed and died very, very quickly, which sounds like a good model for looking at poor patients, but it was probably likely that the pathology that's causing the death of that mouse isn't the same as we're seeing in people.
Yeah, it was very different. You'd see an increase of viral load, an infiltration of the brain.
Okay.
Central nervous system, which is different than what causes mortality in humans. So that's one part of the struggle in transgenic mice. I've got a question, John.
Yeah.
This can go back on the heel of what you've shown us in your presentation. You've shown so many different mouse models, and in your reliance on the mouse model, you would knock out or discard parts of the immune system that you think is important. You mentioned in your brief a few minutes ago that RAG mice were used, RAG1 knockout mice were used, and we know that these types of mice do not have a T or B cell population.
Yeah.
T and B cells are important as far as we know in the viral infections, specifically in COVID-19. So we've knocked these out of these mice, but we still don't see a severe infection. Where does the variation fall?
I would just imagine that because they have not got the right route to get in, so without that kind of ACE2 receptor, they're not going to be able to get in. They can, again, well, I know that the SARS can get in via CD26 as well, but to a much lesser extent. So obviously in the case of the mouse without ACE2, even though CD26 is present, it's obviously going to be a different form. So I think it's in the cases of those mice, it's just not, even without an immune system, if the thing can't get in to start replicating and cause damage, you're just not going to get any sort of kind of model at all, really.
So then how useful are these mouse models then? Are they useful for anything?
I think the ACE2 one is a good kind of model, but I know that people have started to look in other systems, and I saw a paper the other day when I first started looking at this, because I was like, oh, should I get this in? Should we get one of these mice strains in and start to sort of look at it? Because at Lancaster we've got a category 3 mouse system, which is quite nice, but I think it would be fine in Cat 2 anyway, because it's, although it's a nasty thing containment wise, it's not too much. But I know that ferrets, which are obviously used for kind of influenza studies, they do get infected quite nicely. And I actually just saw a paper recently in one of the science journals, which had looked specifically in different animals for kind of levels, and they found, going back to the things you might have seen on the news and random scare stories about cats, but cats are susceptible to it. Ferrets are susceptible to it. Dogs aren't. Interestingly, pigs aren't, which you would imagine would be if kind of, ferrets and humans are, because pigs are a bit more similar. But so there's kind of some strange things there. But there's no evidence that cats are, potential going to transfer it to humans and such. But it's just one of those kind of science-y fiction things that cats could be spreading it. It's got at least some slight basis behind it. But if I'm not a cat or a dog person, so.
Is it? Would you say it falls down to solely the ACE expression, maybe the other receptor you mentioned? Do you think cats express it more than pigs, maybe?
I don't know about if it's the expression levels or the type, but I do find it strange that pigs don't get it and cats do. But I'm not an expert in the kind of molecular structure of those molecules.
So when I read these papers, and I advise all the audience to go and check Sutton et al. His paper in virology, a nice review over the different models of different animal models against the beta SARS or beta coronavirus members, where he lists all these animals that we've just mentioned, the lab animals. Are we now missing the point when infecting these animals? Because I mentioned earlier how in some animal models, we see an infection that spreads systemically to the brain and the central nervous system. And now it's evolving slowly that maybe COVID-19 is not strictly a lung infection. Are we missing something here?
So that's kind of, so I think what people have obviously naturally done is gone to what they know with kind of SARS and MERS and how they kind of really do cause pathology in the lung. I'm not saying that there is obviously huge lung pathology, but just from talking to colleagues who work directly in the NHS and are seeing patients, one of the real standout things that they see are changes in blood clotting. So they see that these people are basically kind of super clotters, this kind of hypercoagulation, and that is occurring in different tissues. So sometimes that, this is talking to colleagues who are working on individual patients. So this could just be one person, so don't take this as some kind of big study, but it was just interesting that they were all saying that there was alterations in kind of clotting ability, and that clots were getting stuck. You would definitely get a lot of clotting in the lung naturally, but they're also seeing things in other tissues. So sometimes kidneys and things like that, where they were also seeing kind of problems. So my question there is, it could be that this is a kind of blood clotting disorder, which is happening, is the pathology you're going to see in the lung, rather than the lung itself being the main site where the problem is happening, if that makes sense. So it's kind of that kind of chicken and egg thing, but I know that there are some studies now looking at kind of big clot busting drugs to kind of use, but again, some of these drugs aren't particularly nice in the first place. So again, it's that kind of end of care stage where people are trying these things.
So on hearing what you were saying, we do see an increase in D-dimers, which are coagulation factors in ICU patients. And I remember posting this question to Marcello Albanese in his webinar, the gentleman who just asked you the question.
Yeah.
And I was kind of torn between whether it's a sign of internal damage and therefore, rapid coagulation factors trying to heal the injuries within the alveoli, or is it associated with the comorbidities that lead to mortalities in severe patients like heart disease and heart failure and hypertension? So does this infection push the comorbidity towards tissue damage? So it's interesting that you're mentioning presence of coagulation factors in the blood, because a lot of papers have been associating it with the severity of the disease, along with other bacterial markers as well. And on the heel of what you were saying as well about other organs, there's a lot of papers coming out about issues in the liver. I don't know whether you can highlight on that a little bit.
Yeah, so I mean, I don't work on the liver so much, but again, some papers have shown it kind of can attack endothelium levels. So again, key organs that it would hit, you could easily see different tissues in that respect. But obviously being biased, I'm kind of quite interested in what's happening in the gut, because I think there's some interesting kind of topics there. And again, one case being that, although I said the lung, it might not be the main site, obviously the lung has the highest level of these kind of ACE2 inhibitors, so it could be getting in there, causing problems in the blood, which the clotting could then go back to the lung. But the gut epithelium is also very, second level of kind of ACE2 kind of richness as well.
Yeah, so Ing Hammings, in his review, he used tissue staining, so immunohistochemistry, in highlighting the distribution of ACE2, and the gut was highly expressive of ACE2, as well as most tissues. Now, he was not able to detect the expression of ACE2s on immune cells like T cells and tissue-bound macrophages, but other papers have now shown that and have have come forward with some evidence to show the expression on T cells and microphages in particular. But that then takes us to one of your other important points you wanted to highlight about the gut and this infection and comorbidity with IBDs. What can you tell us about that, John?
Yeah, so again, the gut thing is obviously my sort of feelings is always it's good to look at different areas with your perspective and knowledge, but I don't think everyone should kind of jump on, I'm not a virologist, I'm not going to be able to answer the questions that they are trained to do, but I can kind of look at the perspective from my, knowledge base. But I know that things like SARS traditionally had quite high levels of gut problems. So kind of around 80% of patients with SARS also reported, intestinal problems, diarrhea and such, and blood in stool. But with the current outbreak, it's a lot more reduced. So there are cases, I think one of the first cases that came in China, the person had diarrhea previously, but it's a much lower level. So it's kind of not been, a big warning sign. You're not told at the moment, at least in the UK, that it's one of the symptoms. We just have this kind of fever and dry cough. Well, diarrhea isn't really mentioned.
The first two cases in the United States had diarrhea and vomiting for 2 days before he was admitted to hospital.
Yeah.
And these stool samples showed a positivity for SARS-2 2 days post symptoms, and it lasted for 22 days. So,
I mean, that's the really kind of, again, with all of these kind of things, it makes you worry, when they say it can survive on a surface for so long. It's a virus, it's not really surviving. You need to know if it's infective. You might find levels of it on a piece of plastic 3 days later, but that doesn't necessarily mean it's effective. But it goes through to this kind of possibility that there could be kind of fecal oral route of this kind of pathogen.
Which supports what you were saying, because Xiao and colleagues in gastroenterology, the reports in gastroenterology have been amazing. I advise anybody and everybody to go and check the reports in their SARS-CoV-2 edition. 50% of people showed, 50% of people who were infected or came or admitted to the hospital, 50% of them showed a positivity in the stool.
Yeah.
And 25% of those 50% still tested positive in the stool, even after the clearance of the nasopharyngeal swab.
Yeah, which is worrying.
Yes,
discharging people and things.
And what's even more jarring is another study showed that 2 people out of 4 or 5, after they showed a positive sample in their stool, and then they were discharged from the hospital after a negative return, so they managed to not show any positivity in the stool. They returned back to the hospital within a week with positive stool samples.
Yeah.
And that's where the journal itself was suggesting the oral fecal route of infection being possibly the main source of reinfection here.
Yeah, it's kind of worrying again, but it's like, is that actually infective or not? The facts. So again, I know from kind of colleagues that there's a massive reduction in the number of cases of V&D, so vomiting and diarrhea at the moment, because people are washing their hands as they should. So
is that an overall though?
Yeah, so yeah, it's hugely down. Like, this is normally a time when you do get it quite a bit, but basically it's really down. So again, just my kind of thing is like, should you order takeout food, if it could be in that route. But I think if we did see that it's kind of fecal oral route is possibility, I think you'd see a lot more outbreaks, and you'd be able to, epidemiologically, you'd be able to trace them back to certain places, whoever was making the food, in a kind of is it Typhoid Mary kind of classical way, if that makes sense.
Yes. So I've got a comment on that, but I'll allow Tarifa to speak.
John, so I just, I want to brush on that. So talking about the virus being viable in the stool, so it has passed through the gut and survived the, the HCl and the low pH of the gut, and it survived until they found that the live virus is in the feces after mostly the symptoms of the respiratory is finished, like the cough and the fever is going down. Then we see the live virus in the feces at day 11.
Yeah.
So the thing I wanted to highlight, I think you wanted to talk about this, is the gastroenterologist in worldwide. They are really concerned about how IBD patients are gonna survive COVID-19. Are they more susceptible because they have high expression of the intestinal ACA2? Do they have to stop their medications? If they trigger the anti-cytokine, level to control their hyperinflammatory, in these patients, are they gonna survive? Can you please tell us more about this?
Yeah, so obviously again, IBD is something I work on. It's a hidden, disability. You wouldn't necessarily know that people have it. It's a young age of people who, should be in the prime of their life, but they've got this kind of horrible thing. And you're exactly right that we said that the gut was a high expressor of ACE2, but actually, specifically in Crohn's disease, when you get inflammation bouts, ACE2 levels have actually been shown to increase. So again, these patients who have got inflammation, they've got higher levels of a receptor which the virus could potentially use. As you say, Tafa, they're on immunomodulatory medicine, so it's very worrying. They're also not having their treatments in terms of colonoscopies for any flare-ups and to check, because those are deemed non-essential and they're just not happening. I can tell you that from looking, because I've kind of got personal interests in looking at why this would be, if this would be worrying, but in China, from their IBD cohort, they reported that no patients with IBD became infected. So that's kind of one bit of information. But then in Italy, where we've had this obviously massive, bigger kind of outbreak now, they've also found that no IBD patients have been kind of infected. That could just be luck, but they looked epidemiologically and they reckon that they should have seen at least 40 to 50 cases in that IBD cohort because of the size of it, and they just haven't seen kind of anything. You could even argue that perhaps, setting aside the ACE2 level, perhaps the kind of immunomodulatory things they are on could even be potentially beneficial, if that makes sense, with this kind of cytokine storm and things. If most things are caused by immunopathology, or it could be an age thing. Most IBD patients are young, and we know that it affects older people more. So there's still lots to kind of look at there. But, the first kind of worry that they might be really hard hit, the initial evidence we've got seems to suggest that kind of isn't the case.
But it goes in contrast with the recommendation from the British Society of Gastroenterologists, where they've only permitted endoscopies, colonoscopies for really, really severe cases.
Yeah, it's like kind of things where it could potentially be a blockage or something. They're looking to see if they have to do surgery. So, and
in their recommendation as well, they do say that they do expect that there will be cases of cross infections.
So again, John McLaughlin, who's the gastroenterologist who did my PhD, a lot of gastroenterologists who do scopes have H. pylori infection, just because, he says he used to joke, no one stirs their cup of coffee with the, but it just happens, so there is a big risk of kind of contamination there. So again, with this disease, and if it is shed and is viable, you've really got to start looking at basically, protective equipment for people doing, colonoscopies and such, to the level we have with full face masks and such like that.
John, can I ask you about corticosteroids and IBD infections?
Yep.
So corticosteroids have been shown to be detrimental in treating coronavirus, second SARS virus. Do you think patients, IBD patients, Crohn's or ulcerative colitis, who are on corticosteroids or any form of steroid are more at risk because of that?
Potentially, I think there's so much going on to play here, it's hard to know. But yeah, I just go back to those IBD studies that from the amount of people they've looked at, if they were an at-risk group, you would expect them to be coming through. Similar with, again, chatting to colleagues in the NHS, people with asthma aren't so, you'd think they'd be one of the big groups, but it's not so much the case. It's more people with kind of underlying kind of obesity, the diabetes, and pregnancy as well are the big things that are jumping out in kind of, again, this is just people, small numbers from colleagues who I've spoken to, but they're all kind of saying similar things.
So, can I add, sorry, Mushraf, before we move? So just comparing corticosteroid to the single cytokine inhibitors, like such, like the trials that are going like IL-6, anti-TNF. So there was that review about controlling inflammatory diseases and treatment for COVID-19 in Nature, like I think 2 weeks ago. So it has highlighted an important target for treating the COVID-19 just by inhibiting the JAK2 pathway. Can you please elaborate on that, because I think it's really interesting that they have looked at different if, like, for example, if they block the IL-6, they will control the hyperinflammatory response, but they will not ensure viral clearance, if you know what I mean. But then the JAK2, it does help with the viral clearance because it does control the action of interferon gamma, type 1 interferon, and JAK1 and JAK3 as well, please.
Yeah, again, it kind of goes back to what is really causing the problem. Is it the virus load that's causing pathology, or is it your own immune response causing the immunopathology? Again, that's something we just don't know at the moment, and we need to kind of find out, again, using good kind of animal models, because there's things that at first instance don't make sense in kind of bacterial lung infections. So you can knock out certain cytokines that are essential or really important in killing off certain bacteria, and you'd be like, well, the animal is obviously going to be worse. But the viral load goes up, but it's just tolerated a lot more, and the mouse then clears it slower, but in a much more controlled way without pathology. So I think those drugs that you mentioned, it all depends. We still don't know, we could knock down certain things like IL-6, and that might be great if it is immunopathology that's key, but if that then allows, anything we come in and suppress in terms of cytokines might allow the virus to boost. And if the virus is the problem, that's bad. But obviously, if it's the immunopathology, that's good. And I think that's why we start to, especially as numbers start to level off, at least in the majority of places, we need to sort of do those studies in kind of animals, to get kind of a good idea, really.
How worried are we about secondary infections when we bring down cytokines?
Yeah, so that's the other thing. It kind of goes to the kind of lockdown thing as well. What is, the difference that we can be seeing there. We might all be safe from COVID-19, or a lot safer, from locking down and things like this. But again, there's been big increases in heart problems. So people have been having small heart attacks. They've not wanted to go to the hospital because obviously they're scared of going and they're feeling I'm not too bad. And they've been going in admitted a lot later when things have not improved. And by then, there's so much damage that's gone on. It's almost too late in some cases to help them. So it's kind of having that kind of balancing act. So again, we could all have take these fantastic things to dampen our immune system if it was immunopathology, but then we're opening ourselves up to other diseases. That's a classic thing with anti-TNF studies originally for kind of autoimmune diseases. People started to use anti-TNF, and then some people died because they had TB underlying. Once they lost their TNF, they were no longer keeping it at bay, and it flared up and was in some quite tragic cases. And now everyone is tested for that before they receive anti-TNF treatment.
Tarfa, can I ask one question as well?
Yeah, sure.
To John? So you were saying about the tolerance, the tolerogenic effects facing a viral load or a bacterial load in animal models. What can you tell us about the involvement of TGF-betas and IL-10 in susceptibility and resistance in COVID-19?
Yeah, so I mean, especially with Werner listening, I won't mention too much about it, but I know for a fact that when they've looked at the kind of cytokine readouts, it does look very cytokine stormy. There's lots going on. You do see IL-10, TGF-beta. It kind of comes to another question of kind of what is the T-cell subset response. So we know that T-cells overall in the sick patients at least seem to drop right down. The ratio, we seem to lose lots of regulatory T-cells, so we lose that kind of suppressive arm, but then we get a lot from the CD8s and the CD4s producing a huge amount of cytokines. We do see raised IL-10 there. I've not seen too much on TGF-beta. Again, it all depends how you're measuring it, because from my work on TGF-beta, it has to be activated, and in a lot of assays, you could have a huge amount in a tube that isn't active, so it's not going to do anything, but in the assay, you acid activate it, and then you give yourself a huge false readout of what's really going on. But I know there is also a lot of IL-10 being made from these patients. I imagine that would be, cytotoxic T cells trying to balance themselves. They're producing huge amounts of gamma and such, and they're probably trying to balance it with 10 as well, and then it's just not enough to kind of rescue it.
Yeah, so that was what Werner was saying last week when we had Joe on the webinar, where he said macrophages produce them, but it's just too much too late in the face of the infection. So it's a sign of deterioration of your body's basically final few breaths to take control of the immune response. I've got a question about the...
I'm glad I said something right about IL-10.
Yes, you did. I want to ask about the T-cell subsets.
Yeah.
And before we go through the T-cell subsets, John, I want you to bear with me as I briefly put in my understanding about what's happening, where you see it appears to be the timing of the T-cell generation is key in fighting the infection. If you fail to develop lymphocytes early in the infection, facing the huge viral load, you will most likely end up with a severe condition. Unlike when you develop it later, after the virus has surpassed its surveillance and managed to infiltrate the body, your late response will be succumbed by the virus, which we don't know why, leading to lymphocytopenia. And you'll have cells that are just chucking out huge amounts of pro-inflammatory cytokines and getting exhausted. What are these cells? There are a few papers talking about Cd4 and Cd8 and their different subsets, but can you elaborate on that for us?
Yes, I actually read, just as you described that, I read something, it was a phrase in one of the papers where it kind of said, people are trapped in the innate immune response. So if you become trapped in that innate response and you don't get your adaptive happening soon enough, just as you said, then when it does kick in, it's too late and seems to be really bad in that respect. So I know people have looked, they can see CD4s in some cases are there, but the CD8s seem to be, so in all cases in these severe patients, the T-cell numbers go down, but the balance, you seem to end up with more cytotoxic T-cells. But again, everything we've got so far is based on kind of just cytokine soups. Some people have done some nice flow experiments, but I don't think I've seen any, even in the nice kind of Nature paper, did they do any kind of intracellular cytokine staining in that one? There's so much coming out at the moment, it's hard to keep up.
And that was one of the comments I was going to bring up, actually. And I'm sure Tarfa had it on the tip of her tongue. I've not seen invasive, well-characterized flow cytometry plots.
That's true.
Because when you want to describe a condition of lymphocytopenia, you need to identify where the lymphocytes are falling. Is the depletion in the plasma, or do you see high infiltration in the tissue? Because you might not have a lot of lymphocytes in the tissue, but you will have huge infiltrations in the plasma and vice versa. So how are you describing lymphocytopenia? And also the markers they use for CD8s and CD4s, they appear to be quite basic as far as we know.
I suppose the thing is, it's, you know, everyone could have their dream study and get things right, but people are really just, the papers we're seeing now are probably literally the first things that people would do. It would be like, right, we've got this panel, let's just stain for what we usually look at and then find out, because I don't think diagnostically people do intracellular cytokine staining as a standard. So that's going to push that window down until you can get research in. There's going to be barriers to get that research allowed and things like that. So I think we'll start to see those bigger papers coming out soon in terms of where they've had a more detailed look at those kind of tissues. But again, that's also an aspect of the lockdown where research labs have been basically told to stop their research. So obviously, I don't know the case at different places, but at Lancaster, we have researchers working on diagnostics and on a vaccine, and they're still allowed to work, but the majorities of everyone else isn't. But again, it's that kind of limbo that if you were in work anyway, you probably would start to go down that route. But when people are, you know, not allowed to go in the first place, then it's hard to get that kind of work started, if that makes sense. But there's no kind of, that's not a go at any government policy or anything, you know, that's just, they're doing what seems to be best at the moment.
All right, so drum rolls, because I think we've got Joanne Perrock who wants to participate. All right, Joe, can you hear us?
Hi, John.
Hi, Joe.
Hi, John. I'm good, thanks. Nice to see you.
Good to see you. I can't see you, but
yeah. I'm not. I might. Shall I do my? Here we go. I'll say hello in a minute.
Oh, yeah.
Hey, Joe.
I'm going to stop my video because I'm on Welsh internet and it's really slow.
No,
but I just wanted to chip in actually, because in Manchester, Tracy Hussell and Lizzie Mann are doing flow samples, really big panels on COVID-19 patients, and they shared some of their preliminary data with us today, and they're seeing the lymphopenia that everybody else is seeing, but they also see a huge increase in monocytes, which not many people have reported, and neutrophils as well. And that the reason they're struggling with their panels is because they can't get the antibodies delivered. So they're stuck with the ones that they've got in the fridge and the intracellular stuff that they've got. But I think there'll be some quite nice data coming out of Manchester, because they've got a spread. They're doing longitudinal studies across sort of a month. And they've got a mix of severely ill, moderate and mild patients as well. But something that they do see, which is really interesting, is that B cells and lymphocytes, the proliferation goes down initially, but as the patients go into ICU, they start to accelerate and come up again. And they're wondering if that's to do with the possibly secondary infections.
Yeah, it could be, because that is the thing that basically it seems to be, most people, it's when they come in to ICU, it seems to get worse, basically.
Yeah, exactly. So I think given time and delivery of antibodies, I think there'll be quite a few labs that are going to hopefully get some of the basic immunology out, which I think will be great. Yeah,
you kind of know that places are doing it. It's just that kind of...
could do it. Yeah, exactly.
Yeah, it could do it as well. But like things like deliveries as well,
Yeah, no, it's hard,
So Joe, can I comment on what you were saying? So there's a lovely review in Nature Review Immunology where they suggest that the target for treatments should be the myeloid cell compartment. So that's on the heel of what Tracy Hussell showed. Has she looked at any cytokines? Do you know?
So a little bit, and I know that the data really is preliminary, but one of the interesting things they've seen in their patients who've all come from Salford Hospital is that the peak in TNF drops quite quickly as patients go in, become more severely ill. So, exactly as John was saying, with these IBD patients, a lot of them are on anti-TNFs, and I've read the same thing as well, that there are far few, and that's been reported in Manchester as well, anecdotally, that they were expecting to have all these patients on immunotherapies to pile into the wards with coronavirus. And actually in Manchester, in the children's ward at least, they've converted their sort of immunotherapy ward into an ICU because they've had so few patients in. They were keeping it to one side and then the patients never came. So we definitely need to find out more about the anti-TNFs and the other immunotherapies that are out there for sure.
Definitely.
The best time to give them, really.
Joe, may I ask you, the study that Tracy is doing, did she consider looking at the T-cell count, CD4 and CD8, in those severe compared to mild group?
Yeah, so that's one of the difficulties, I think, because they're getting, it's the age-old problem, isn't it? At the moment, they've got percentages, and they're trying to convert that to counts given the samples that they've got. So I think that data will come out, but it was just looking at percentages today. And you could see that, as John said, the CD4, CD8 compartment goes down, but the ratio is much higher within the CD8s. But they also show a really significant drop in NK cells with severe patients as well, which was interesting. So as you can imagine, John, Dan Davis was jumping up and down in his seat. He's an NK specialist for everybody else. Okay.
Well,
thank you, Joe.
Thank you, Joe. Thank you very much.
Good to hear from you. Yeah, so no, I've also seen in a few papers where, again, with lots of, as Joe said from Tracy's work, lots of neutrophilia, lots of neutrophil, like the majority of cells were neutrophils that they were seeing. And again, and I know there's lots of evidence for in the blood. It doesn't look like normal sepsis, though. Lots of the blood samples, the blood's very, very high in triglycerides and things. And again, going back to that kind of clotting thing. So it's not a kind of classical sepsis that you would see. But I mean, the great thing is we have all these different disease models, and we can tune into the things we all see and, come together as a community. And I think that's one of the great things that's happening. I'm talking to you all now, which is a really fantastic experience, to kind of, which probably wouldn't have happened before. I'm sure we're all keeping up to date with the journals, which have all been made pretty much freely available for the COVID side of the research as well. So again, that's a really great beneficial thing as well. So I think we can also, keep in mind and look at the positives of these kind of things.
So just to mention about the Cd4 T-cell, I would really like for you to highlight on this. A study came like last week, dysregulated of T-cell. I think that was a preprint, and it suggested that the problem in the severe cases is due to the ratio of CD4 naive to memory T cell is altered in those severe cases. So they have more naive CD4 cells, but less memory CD4 T cells for some reason. Their CD8 was fine, not significantly changed with the mild group, and the B cell count was fine. So what do you suggest? Like, what's the problem of these CD4 naive cell? Why they're not differentiating? What's the problem? They're not giving the signal to B cell for antibody response in those severe cases?
Yeah, so I suppose that kind of, I suppose it's if you're losing your CD4s, we know how bad that is from HIV and things like that, which could even go back to the secondary infection things that Joe was talking about. Yeah, if you lose that arm of your
immune response. Is that due to lymphopenia in those patients? Do you think this is
I wouldn't know really. I mean, yeah, it could potentially be, but it's interesting that the majority is still in that naive state. That is kind of interesting.
So there's a paper by Lee et al. In the Journal of Immunology, and in this paper he elegantly kind of discusses the lymphocytes and T lymphocytes and the balance between CD8 and CD4 in the first SARS, so SARS-CoV-1, I think, and he associates severity with the drop in CD4s, and he dissociates CD8 completely from the severity of the disease. Does that make sense, John, to you? Because for me, there's a link between CD4 and B cell activation, plasma cell production, and the orchestration of Th1, Th2, and the other T helper cytotypes. While CD8 is this hardcore cytotoxic T cell. And this dissociation for me made sense. Am I missing something here?
No, I think that does make sense. I think you've got to remember that CD8s and what they can produce, you know, they're often looked at as, you know, very one-dimensional compared to the helper T cells, but obviously CD8s can play different roles as well in that respect. And we've not really, I mean, we don't know what's happening with kind of innate lymphoid cells as well. We've not gone down that route yet, or I'm sure groups are, but obviously if it's difficult to get antibodies on, they require a very large panel to rule everything else out as well. So that would be kind of interesting.
I've got a question as well about the homology between the first SARS and the second SARS and MERS. So these two key members of the beta coronavirus. And I'm presenting this question because it's been presented nearly in every webinar we've given. And the question is in simple terms, and excuse my very simple wording of this. If somebody has been able to develop neutralizing antibodies against SARS-1, and homology is reaching up to 78% or 80%, why are they failing to protect themselves against SARS-2? When you are, again, time and time again, saying that the spike protein is our target.
Yeah, I think that, again, that was kind of a sort of a question I heard a little bit earlier on from Wissam. You know, is that the target in terms of this vaccine? You know, it is World Immunization Day as well. You know, so we all hope it works. But if it does work, it could work. But in terms of it could do exactly what people want. It could target that spike protein. But is that the thing we want to be targeting? And that's the thing with, you know, vaccines and such, especially in this kind of not rushed, but this urgent state. You know, you could give that to the patients could make a really nice response. You could get lots of antibodies to that spike protein, but we don't as yet know if that spike protein is the thing that those patients that are doing well are making a response to.
Yes, especially with antibodies. They can go either way, can't they?
Yeah, no, exactly.
I was thinking, I'll give one more question to you, John, and if Tarfa has something to add, then we'll open the question for our audience.
Yeah.
So again, in this paper by Lee et al., where he discusses SARS-1, and the reason I'm referring back to this paper is so many interesting findings here, where he says the CD4 population, the majority of them, are the CD27 positive, CD45 RO positive central memory CD4 cells that are able to mount an immune response here. I found this a bit, interesting to me, because this is a primary infection that you have the involvement of memory CD4 T cells in its fight.
Yeah, I suppose.
What am I missing?
I'm trying to think in terms of there could be some. I mean, that could go with kind of Arnie Akbar's work on sort of senescence as well. So he's always had this state that as we build up different memory cells, you've only got a certain space where you can have that memory, and as more and more things you encounter as you age, certain things get pushed out. So it could be a possibility that we might have some kind of sort of residual or cross response from that. But again, it's always hard to identify the different memory subtypes, from kind of, we have this very effector memory-like kind of markers sometimes. And I'd have to double check the exact panel that they've used. But there's, again, with everything, there's always some crossover where is it a true long memory cell, or is it something that's just been made which has a memory-like phenotype? But again, until we have those more detailed studies, it's hard to kind of answer in that respect. But it could be potentially that there is some kind of cross reactivity.
Tarfa, do you have anything to add before we go to the panel?
Actually, no. I have one question from the audience, if you may allow me, and then we can take some of those who want to engage by asking John directly.
Okay.
So it's from Faisal Minshawi of Manchester. So he asks about if you think that there is other modes of transmission of the coronavirus to the intestine. So how the virus is migrating to the intestine, if there's other routes other than infected immune cells?
Yeah, so I think one of the main things would be, if we avoid that kind of fecal oral route and take that from the side, if it does come in classically through the lung, you do see that thing of, literally people coughing up sputum, and then you will swallow it and it will then go down, and that's how would be another option of kind of infection, and that's been shown with kind of SARS and things in the past. So I would imagine that would be one of the ways that things are happening in that respect.
Okay, so we have Antonella, I think. Yeah.
Antonella?
Hello, can you hear me?
Yes, we can.
Hi, yes.
Hi. Thanks, John, for your presentation and for your enthusiasm about being a community. That's really nice. Yeah, I'm going back to this topic of orophycal transmission, sorry. I don't know if I just missed it, but in SARS in 2002, there was a paper showing cases of transmission through toilet pipes within a compound. And of course, in this case, I think the root of infection can be respiratory tract if you inhale the particles, or also digestive route if you swallow these particles. Either way, I don't know if I'm saying something silly, but in the case the infection starts in the gut, I think these patients showed evidence of respiratory problems in any case later. So how does the virus get from the gut? How does it make its way to the lung? I don't know. I'm confused.
Yeah, so I suppose, you could, with the kind of fecal oral route, if that's possible, you're probably most likely to infect yourself, from that. And then you could potentially, you'll then touch it, touching your eye, touching your face, and then, or get back down the mouth, but also, into the lungs from that respect. Obviously, if the virus gets across, we have some things we've seen, really nicely. So in Manchester, Richard Grensis has shown that helminth infections, that a worm that just lives in the gut, you have responses from innate lymphoid cells which can travel to the lung and then cause changes in the lung, which stops the lung being infected by worms. So again, there's no reason why, if the virus is in certain cells, that they would be able to migrate to other barrier tissues, and then you would have the infection there. So a kind of systemic kind of infection.
Sure, and I was wondering...
there's definitely a gut-lung axis particularly.
There is a lung axis, yeah. No, because I was also wondering in the case of IBD patients, or where there is a problem of gut permeability, whether this would, of course, promote the spread of the virus, and I hope they will look into these.
Yeah, so we touched that on a bit, that we were all expecting, worldwide, that IBD patients and people immunocompromised would really be susceptible, but that doesn't seem to be the case. But there's no reason that, if the first point of call is the gut and you get damage and leaky gut, then that would be a clear way and a very advantageous way for the virus to then travel around the body very quickly.
Sure. Thank you.
Thank you, Antonella. Tarfa.
So do you want to take a Ghaida question, please?
Let's take Ghaida, then we can read a question out of the chat.
Okay.
Ghaida, hello.
Hello. Can you hear me?
We can hear you.
Hi.
Yes. Assalamu alaikum. Thank you for great information. I have one question. Explain the role of immunity system in development of gastric cancer in patient has H. pylori.
Say that again, Ghaida, sorry.
I need to explain the role of immunity system to development of gastric cancer in patient has H. pylori.
Okay, so maybe, if I understood your question, you want John to elaborate on how H. pylori advances the progression of gastric cancer, right?
Yes.
Do you mind if we keep that the last question, so you can stay around?
Yeah.
Okay.
Sure.
Thank you, Ghaida.
Okay. Thank you.
So you stay around, don't go, so we can follow the line of it. Thank you.
Okay. Thank you.
So we'll take. So because it's a very long question, John. So if you can maybe elaborate on that before the end.
Yeah.
On H. pylori and gastric cancer. Thank you, Ghaida. We'll have Rahaf al-Sufi.
Hello.
Hello, Rahaf. So she's one of our students. Go on, Rahaf. What do you have?
Okay, so earlier you were talking about the relationship between leptin and the immune response. So I was wondering, have you looked at the case and the relationship between leptin and COVID-19 patients?
So,
relationship, yes.
Yeah, so I did. I haven't personally, but I literally PubMed, leptin, and kind of COVID-19, and there had been sort of some correlation. I don't know if it was actual samples, but someone had looked very much, they'd related the COVID work with work on influenza. And basically one of the key things is obese patients tend to do a lot worse with influenza, but they also seem to be kind of super spreaders. So with influenza, if you've got especially high leptin levels, you can then, it seems to actually be detrimental there, and you end up having the virus tighter for longer, and it taking longer to get rid of as well. So I haven't obviously looked personally. I've not got any. We're looking at getting access to samples at Lancaster just to look at certain things. Again, we're going to be looking at the kind of microbiome side of things within the gut. But so no one's looked precisely from COVID-19, but there is something in the literature where someone's related the potential, a kind of hypothesis paper to the levels of leptin and kind of obese patients, because obviously that's a big thing. Obesity is a really bad sort of indicator of kind of responsiveness to this virus.
Very good question. Very good question. Proud of you. Very good question.
Thank you.
Joe actually asked that question as well privately on the chat. So you're in very good company there.
Who do you have?
So Asil Hassan.
Okay, Asil. Can you hear us, Asil? Another student of ours.
Yes, can you hear me?
Yes, we can hear you, Asil.
Okay, I want to say, as we read, there are 2 Chinese doctors who felt critically ill with coronavirus, found their skin had changed color. I read some papers that say that it's because of lack of oxygen, and some say that is because of some disease in the liver. Can you explain it?
I can. So I'm not a medical doctor or clinician, but what was the kind of change of color?
What do you mean change of color, Asil?
There was their skin was white, and they become like a brown skin.
Skin discoloration. I've never heard of that symptom.
So sometimes with kind of cytokine storm. So were these patients very severe, or?
I didn't hear you.
Were these patients very severe cases of COVID-19?
Yes.
Was it?
Yeah. Okay. So what happens with cytokine storm sometimes is that if you look at going back to my work, not my work, but my knowledge of regulatory T cells. So in the scurfy mouse, which is a mouse that lacks regulatory T cells, at the extremities of kind of barrier sites, you get lots of inflammation and disease. So the ears of these mice become very inflamed and extremities. And even in patients who trialed, I don't know if you remember, there was a trial of a drug, an antibody for CD28, which basically the idea was it would dampen down inflammation, but it actually did the complete opposite. And these first few volunteers for the ethical study became really, really sick because they had a cytokine storm, and the tips of their fingers actually went brown and black. So that was actually damage caused by the inflammation, and also it can be basically a lack of oxygen getting to the tips. So it could be 2 things there. It could be they were undergoing a cytokine storm, or basically a lack of oxygen, perhaps through damaged lungs, which were getting to the extremities, because those are the first things to kind of go in both lack of oxygen and cytokine storm.
Well, thank you, Asil. Thank you very much. John, can I ask you a question? If I want to take this back to antibodies, I know we didn't talk much about antibodies in this webinar, but we did in the previous webinars. It appears that in SARS-1, people retain anti-spike specific IgGs upon recovery. But then there's another form of antibody assays, which is called neutralizing antibody activity.
Yeah.
What does that mean? What's the difference between having anti-spike protein levels of IgG, and then that's kind of isolated or different than neutralizing antibody activity within the same patients? What are we looking at here?
I think it's just looking at the functionality. I'm not too sure the different medical tests. I mean, you might be able to say a bit more than me on this, but I think it's just to do with the functionality of the actual antibody and how effective it is.
Because I would imagine then, if I have my reservoir of anti-spike antibody proteins, then they would be neutralizing antibodies, would they not?
Well, again, it all depends on what area of the spike. It's kind of classic kind of correlation in things, like going back to the presentation I did earlier, where we saw that CCK cells went up, and we saw that there was a reduction in feeding at exactly the same time. So that was a really nice correlation, but it didn't necessarily mean that one was causing the other. And that's why we had to get a mouse in which we had CCK blocked before we could actually make that conclusion. So you might have lots of antibodies that are able to bind to different epitopes on the viral spike, but it doesn't mean they're going to be kind of functional. And that's the big problem with kind of the vaccine trials and things. You go for the thing that you think is most likely to have the effect, but it doesn't necessarily mean it's going to be the case. Even in a patient, they might have you would always probably go for the most likely, the highest level of a certain antibody, but it might be the fifth one down on the list that is actually really having the effect. And it's very difficult to kind of know what that is. We're able now to do computer modeling and such to really try and predict what would be the best. But so you might have lots of antibody to parts of the virus which is ineffective and doesn't stop it working, but it's finding that key one that actually does the job.
So do you think, another question where I'm trying to stretch your immunological brain, do you think then we identify different epitopes as individuals? So my immunity would detect a different epitope than yours, yet both of us will be resistant or able to mount the proper immune response? I
mean, again, I'm not on kind of antibody sort of function. I'm not an expert.
Not necessarily antibodies. I mean, even antigen recognition in any cell, T-cell receptors, MHC. If I recognize an epitope, what's the likelihood that my epitope or the epitope I recognize is completely different than the epitope your immune system recognizes?
I think the thing is, most of us will recognize a good variety, but we should all. The great thing about the immune system is we should all pretty much come up with the variety, and the one that works will then basically be able to kind of get expanded. So, and again, in populations, you will see some people who will make that very quickly, and they will not really show any infection, while others, it will take time for that one to come through and they'll have a bit more disease in that case. But again, that's very much with kind of clonal selection and things like that kind of comes into that.
Yeah, so on the heel of that, and then we'll have Tarfa chip in. So they've identified 55 and plus epitopes of protein targets.
Okay, yeah.
On SARS-CoV-1 and 2 that have been associated with the development of the proper type of CD8 positive T cells or Cd4. So it's interesting.
So can patients have? Do all patients have that variety, or is it if you get one of those, are you okay?
So what this paper says, and I'm talking about Lee et al. In Journal of Immunology. So spike protein generated the most dominant T cell response, up to 41%. Spike protein itself of T cell responses, and the least was the replicase molecule on the virus. And even though the replicase molecule on the virus, understandably, covers 2/3 of the viral genome, so even though it's vastly expressed within the genome, it's the least targeted by the immune system. Other targets by the immune system that have been identified are the I said replicate, the spike protein, the envelope, the membrane, and the nucleocapsid. So the most successful targets thus far that have been identified are the anti-structural proteins. So spike, membrane, envelope, and nucleotide, or nucleocapsid. So I don't know if that answers the question.
No, I think that kind of covers it nicely.
Tarfa, are you there?
Yes, actually, I wanted to go back to the IBD patient, if you may let me. So after, if there's no one, I think there's no one left to ask questions.
So we'll have to go back to
audience.
Reida's question. We'll go back to Reida's question at the end, but yes.
But just a quick question. So I don't know if you have seen a paper by Greg et al. About IBD patients and novel therapy regarding using soluble ACE2. Did you see that? So theoretically, as we mentioned, I'll just tell you my question quickly so Ghaida can go ahead. So theoretically, we think that IBD patients are more susceptible because they express ACE2 in a high expression because all of the immunomodulation treatments they're going through and
especially
and the inflammation.
Especially Crohn's patients have been shown to have high levels.
But the thing that this study by Greg et al., I think it was released last week, they found that the soluble ACE2 in the plasma of the IBD patients are maybe something that is protecting those patients from the progression of the disease. So they have higher amount of soluble ACE2, which is interesting, I think, because until now, we did not confirm that IBD is a risk factor with COVID-19, if you know.
Yeah, so we could, no, that's really interesting. So we said, well, like we said, we all thought they'd have more, but they seem to be completely, very nicely.
So I think this is a nice approach if they can, and something quicker than the vaccine.
I suppose the idea there is you've got so much of the receptor
in their plasma
binds to floating around that it just mops it up and then it can't. Yeah, no, that's a nice hypothesis.
So, John, let's have Reida's question. So she was asking about how does the H. pylori infection?
Yeah, so basically, from my understanding, so it was the famous kind of experiment where it was very much linked to kind of ulcers and cancer and stuff, and this infection was driving it. People didn't believe, so the guy drank H. pylori, had a bit too much, and basically had massive ulceration and had to go to hospital because he started to cough up blood. So he kind of proved his point in a nice experiment, but he kind of did it a bit too far maybe. But basically cancer is very much a kind of inflammatory disease. So if you get an infection that's causing inflammation, you can have increased turnover, the kind of wrong kind of cytokine signals around there, and that drives then kind of cancer cell-like formations. So there's a model where people use, that we use ourselves, which is the azoxymethane DSS model. So that causes, so if you inject AOM, you give one injection and the mice will eventually get tumors. So it's a carcinogen basically, but that takes a hell of a long time. Even mouse living for, a year might not have any tumors at the end, or very few. So what we do is we give an inflammatory stimuli at the same time. We do an injection and we give dextrin sulfate sodium, which is DSS, which causes colitis. We put that in the drinking water. And then what happens is animals will then basically have this period of inflammation, and what can happen then is that drives the cancer forming cells a lot more. So basically, in answer to the question, if you have infection or anything that causes inflammation, it produces a more cancerous state, because cancer is kind of known as an inflammatory state in itself.
Well, thank you for that, John. Tarfa, do you have anything to add?
No, I think there's no more questions, I think, from the audience, but that's it, I think. I can't see any hands.
So I've got one question, John, from one of my students who was saying, so I'm translating this from Arabic, if we target single cytokines, how effective will this treatment be when we are facing a cytokine storm?
No, exactly. We can take one player out and then something else might come in to play a role in that respect. So I suppose we would have to look at the kind of sepsis studies there. Sepsis is still one of the biggest killers. Sometimes patients aren't necessarily, we don't know that they've got sepsis. It's not kind of classified that well. A really nice recent study actually kind of tried to estimate the numbers, and it's a huge killer that we don't really take into account. And we're still having to find methods of dealing with that. So it's not going to be necessarily the case of taking out one particular thing. You know, some things go hand in hand, like, you know, IL-6, TNF-alpha, but it's not going to be as simple as that. If it was a simple, you know, block 1 cytokine, we would have probably done it by now. So no, it's a good question.
We would have treated autoimmune disease for one. If we would have managed. Okay, I think unless Tarf has something, I think we've managed to get spot on time, John. For you to catch your tea. I'd like to thank you for giving us this opportunity. I hope you found this stimulating as we did.
No, it's great. Thank you very much for the invitation.
Anytime, and hopefully we can do more of this. The majority of the attendees were students and a lovely group of my colleagues. This is the last webinar episode, so it's my honor to have one of my best friends to end this on.
Thank you very much.
So any final words you want to give to the young students who are hearing you now?
No, I'd just like to say thanks all for listening, and hopefully you found it interesting, and also some really nice, clever questions that I got from you all as well. So, and again, just kind of enjoy the sort of togetherness of the immunology community. You know, done really novel idea from Mushraf to get this going as well. So hopefully it's been kind of stimulating for you all, and just wish you kind of a happy and kind of safe Ramadan as well.
Thank you, my friend. Thank you. Tarfa, thank you very much for helping me today.
Thanks a lot. And thanks a lot, John. Thanks a lot for a nice talk and discussion.
All right. Thank you. Thank you very much, all of you. And hopefully have a great Ramadan. Ramadan Kareem lakum kullakum jami'an. And thank you very much, John.
Thank you.
Thank you. Bye-bye, my friend. Thank you.
Okay, bye. Cheers.
Bye.